Hello, everyone, and welcome back to the Validity Podcast. I'm your host, Valentina Rodriguez, and this is officially episode 2 of season 2, and it's the first one where we're going to get really into some actual science, given that it's now been 2 weeks into the semester, so we finally started to cover some actual content. And as I mentioned in the previous episode, most of the content that I cover in the season, at least from the semester, is going to come from my neuromuscular exercise physiology course. And I told you guys last week that this week's episode was most likely going to be on the more molecular side. And so indeed, this past week, we were lectured on the molecular response to resistance training. So we're going to talk a little bit about how we actually adapt. So, we're training, we're doing stuff at the gym. What is happening at the molecular level in these muscle cells, in these muscle fibers, to eventually result in an increased accrual of muscle mass? Like, what exactly is happening? What is the pathway? And we don't fully understand it, not 100%. We don't fully understand the mechanism for adaptation to resistance training, for muscle growth, for strength gain, but there's different theories, and we're going to discuss some of them. I will say a lot of this episode, especially when we get into the actual signaling pathways, will probably require some basic biology or even like intermediate biology. I'll do my best to try to explain concepts to the best that I can, but I obviously cannot give like an in-depth, like basic or intermediate biology lesson before we get into this. So I'm gonna do my best to make it as digestible as possible. But before we get into the science, let's just do some personal updates. I am now about 6.5 weeks post-show, hovering between 3 to 4 pounds above stage weight. So I'm sitting in a good place. It's still a mind fuck to watch myself get softer. But I was talking to one of my friends who's also kind of reversing right now. And sometimes what you just have to do is like wait it out, because your brain literally changes the way it perceives your physique when you get that lean. And the way that I look right now is completely fine and fantastic. And I know I look lean and great and whatever, but my brain not able to perceive it in such a positive light because it's still being warped, so to say, from being so stage lean. And sometimes it's just a matter of time and just existing in a softer physique to let my brain just get used to that. And my physique itself does not necessarily need to change. Like, I objectively look great, but subjectively my experience and my perception of my physique is still not in the most positive place. And it's just a matter of time and a matter of like adapting and getting over that stage lean mental hump. And like I said, it's not that like my body needs to change for me to feel better about it at all. It's just a matter of my mind changing, and I'm working on that daily. I am still taking 1 to 2 free meals a week. Food is high, cardio is low. Food's been going up every single week. I'm trying to figure out what I'm going to do for next week's episode, because next Thursday I leave for a wedding, and that means I have to have the episode pre-recorded and edited before I leave for the wedding. So anyways, that's a me problem to worry about. And the only other update I have is really that we're still recruiting for the world's first reverse dieting study as a part of Dr. Campbell's Performance and Physique Enhancement Lab. I am leading that study. And if you are interested in being a subject, it will entail a maintenance, a dieting, and a post-dieting phase. So if you are interested or just want more info or want to see your eligibility to be a subject, please email reversedietstudy@gmail.com. And we'll get back to you with more info, eligibility forms, etc. And then other than that, we're literally going to get straight into the science. So this entire lecture that I'm going to be recapitulating to you guys is the molecular response to resistance training. So essentially how we adapt, how the muscle adapts to resistance training. Because at the end of the day, building muscle is your body undergoing a physiological adaptation to the stressor of resistance training. You place a certain load, you create a certain stress, and there is a signaling pathway. There is an adaptation response that results eventually in bigger muscles or more strength in order to theoretically be prepared to take on that stressor next time. Theoretically, like I said, a lot of why we adapt and the mechanisms to adaptation are theoretical. We know the pathways, we know how they work. Those are not theoretical, but it's still theory exactly how and why we adapt. So I want you guys to keep that in mind. So essentially, adapting to exercise is you resistance train. That serves as some kind of signal that's sensed by some sort of sensor protein. Those signals are conveyed and computed by a signal transduction pathway that eventually results in the creation of more protein, more contractile protein, more sarcoplasm. We don't fully know with confidence what exactly the end result is when we train, but we do know it results in bigger muscles and more strength depending on what exactly your training was. And when we think about why we adapt, a lot of the whole progressive overload and the whole theory of adaptation comes from a man named Julius Wolff, and he's the one who created the overload concept. And he created this, or he came up with this, or he theorized this based on bones and mechanical overload. And this theory was extended to other organs, but it explains kind of why we adapt. Like you overload, you stress an organ, whether that be a bone, whether that be a muscle, because it underwent the stress, it theoretically needs to be prepared, like I said, to take on that stress next time. So it adapts in order to be better prepared, better suited to take on that stressor. So it might be a good theory to explain why we adapt, but we still don't know how we adapt. Still doesn't explain the mechanism. And so there's a different theory out there. It's called the supercompensation theory, and it's really closely tied to the general adaptation syndrome, which I'm pretty sure we have an episode on when it comes to periodization and all of that. So go back and listen to that. But essentially it kind of says that you need to like recover. You train, you stress, then you recover. So you pull back, and that recovery causes you to overshoot above baseline. The response is an overshoot response. It's an extension of the general adaptation syndrome. And the fundamental flaws that we talked about when it comes to the supercompensation theory is that we do see supercompensation for muscle glycogen after endurance training and then after feeding. But the time course for the supercompensation effect when it comes to muscle glycogen, so you deplete muscle glycogen, you feed, you actually end up with more muscle glycogen than when you started with. That's supercompensation. But we don't see that in mitochondrial biogenesis. We don't see that in the creation of new capillaries. We don't see that in the creation of new neurons. Like, you don't lose mitochondria before you create more as an adaptation to endurance exercise. So when you do cardio, you undergo something called mitochondrial biogenesis. It means that you essentially, if you train and you do cardio and you do a lot of endurance exercise, you're going to increase the number of mitochondria in your cells. And mitochondria is, we all know, the powerhouse of the cell. It's essentially where a lot of our energy production processes are housed. So more mitochondria means more capacity for energy production. So it's an adaptation to help you sustain the endurance exercise that you're consistently putting your body through. But when we see that adaptation, when we see an increase in mitochondria, we don't see a decrease and then an increase. So we don't see a supercompensation as a response to like pulling back and then overshooting. We don't see that in the creation of new capillary networks. We don't see that in the creation of new neurons. So yes, supercompensation can be applied to like muscle glycogen, but it cannot be extended to other organs and other types of adaptations. Another flaw is that it proposes a time course again, but not a mechanism. It still doesn't explain how that adaptation actually occurs. Like, what is the pathway behind it? Another flaw is that it also implies that recovery is essential for adaptation. And don't get me wrong, recovery is really, really important, but it might not be absolutely necessary critical for adaptation, at least not when we talk about cardiac muscle. When we think about the heart never rests. And if you do enough endurance exercise, you do undergo cardiac adaptations. You do undergo thickening of your left ventricular wall. You do increase your stroke volume. You do undergo adaptations of the actual cardiac tissue, of the actual organ, the way that it functions, the actual morphology of the heart, the size, but without any rest. Your heart never stops beating. There was even a study done by Henriksen et al. Where they did chronic stimulation of a muscle without rest. It was muscle stimulation. I think it was 30 hours, 3 times a week for 5 weeks, and it adapted. It grew. That was without rest. So those are 3 fundamental flaws of supercompensation. And again, all of this explains a potential theory of why we adapt or necessary requirements for adaptation, but it has some flaws that we see adaptation in other organs that don't undergo the supercompensation. And also it still does not explain how we actually adapt. So there's another theory for adaptation, and this is the signal transduction hypothesis for adaptation. And this is the more molecular one that we're really going to be looking at when we get into the actual pathway of what happens when we resistance strain. So this overview of this signal transduction hypothesis for adaptation is that there's specific sensor proteins that detect your exercise signals. Those are, again, computed into signal transduction pathways. Those signals result in an increase in gene transcription and translation. I'm not going to get into transcription and translation. So if you need a quick overview on that, it's just essentially turning your DNA into mRNA, and then mRNA provides the code, so to say, for protein, and ribosomes translate the mRNA into functional proteins. So again, you exercise, sensor proteins detect that, they compute that into a signal transduction pathway that results in gene transcription, gene translation, and then that results in protein synthesis. And the overall result of that protein synthesis is some kind of change to the organs. So increase in muscle mass, increase in strength capacity, et cetera, et cetera. That's just an overview. And we're going to actually be talking about what the key players are in those signaling pathways. So that's what we're getting into today. So this episode, I'm going to just give you like the basic fundamental molecular biology of what happens when you resistance train. And what I just kind of went through was hopefully helping you contextualize all of that within the existing theories of adaptation. So the basics is we have genes. Every single cell in your body has a nucleus, and within that nucleus, you host your entire genetic code, your entire DNA, all of it, in every single nucleus of every single cell in your body. Genes are stretches of DNA that encode for proteins, and there's certain steps that need to occur in order to go from DNA to functional protein. The stimulus of resistance training itself activates genes. So when you resistance train, there is an upregulation in the transcription of certain genes, one of those being mTOR, and we're going to get into that later. We're not going to actually talk about what happens between when you lift the weight. So what happens between that one rep, that one bicep curl, and the actual increase in transcription of a gene. We're not going to get into that. We're going to start from when that gene is activated, how that results in an increase in muscle mass. Okay, so we're going to kind of take the latter half, because the first half, the former half of that entire pathway, how you go from resistance training to gene activation, is not fully understood. But we know what happens when we have a gene whose transcription has been upregulated. We know how that trickles down the signaling pathway to result in an increase in muscle mass. So that's what we're getting into. So like I said, mTOR is probably something that you've heard about. It's something that maybe comes up in studies, because I know last episode we talked about how different measurement tools are important in being able to adequately convey the results based on your study design. If you do like a muscle biopsy, you'll be able to actually measure levels of certain proteins, of certain molecules, of certain biomarkers that are indicators of either an increase in muscle protein synthesis or not. And so I think after today's episode, you'll be able to read research that looks at those biomarkers, that reports changes in mTOR and S6K and different, like I said, molecular players. And you'll be able to understand why those are indicative of either increases in hypertrophy or not. So again, we have genes. They're stretches of DNA that encode protein. When you train, those genes are activated, and those genes are transcribed into mRNA. For a gene to be expressed as protein, the genes must be transcribed into RNA. RNA polymerase is going to scan the DNA, transcribe DNA into mRNA, and then mRNA is going to be processed. It's going to be spliced by spliceosomes, and that mRNA, when it's ready to be turned into a protein, there's going to be a whole bunch of things that have to happen and come together for the ribosome to eventually be able to translate that piece of mRNA into protein. And when we think and we talk about muscle protein synthesis, the major rate limiting step, the major component of this entire process that determines the rate at which muscle protein synthesis occurs is translation initiation. Because like I said, there are a lot of moving parts that must be put into the right place before ribosomes can start turning that mRNA into functional proteins. And we're going to talk about what those key players are, what those moving parts are, because they are what's either increasing, decreasing, just determining the rate of overall muscle protein synthesis as a response to resistance training. Overview, you have a gene in your DNA. It's a stretch of DNA. That gene is activated. RNA polymerase transcribes it into RNA. It gets processed, spliced, becomes mRNA. mRNA then is ready to be translated, and a bunch of moving parts have to come together to create the final functional ribosome that's going to translate that mRNA into a functional protein. So let's talk about mRNA translation. This is the primary process necessary for the development of compensatory hypertrophy. And your net muscle protein synthesis is sustained via the upregulation of translational machinery. So you increase the efficiency or the capacity of translation, you increase your rate of muscle protein synthesis. The mechanism of muscle protein synthesis includes, again, the translation of mRNA, and it's also combined with post-translational processing and the transport of polypeptide products, but we're really going to talk about what determines the rate, that rate limiting step, which is translation initiation, efficiency, and capacity. So in the short term, you get an increase in muscle protein synthesis from the upregulation of mRNA translation. So more RNA translation, more muscle protein synthesis. But in the long term, if you want chronic upregulation of muscle protein synthesis, that needs to be accompanied with an overall upregulation of translational machinery. So think about like in the short term, your ribosomes, your translation machinery is working faster, and that's how you increase your muscle protein synthesis, or your MPS. We're going to use that acronym moving forward. So in the short term, you increase your MPS by just making that machinery work faster. But in the long term, it can only work so fast. So if you want to increase your MPS over a long period of time chronically, then you need more of that machinery so that you can get things going at the same time. In class, we talked about, like, say you have like a McDonald's and it starts to get really high demand. So you start cranking out burgers really fast. But say the line starts to get really long and there's only one McDonald's for the entire small town or whatever. The town starts to grow and people are like, we want more burgers. That one McDonald's can only crank out burgers so fast. So you might need to build another McDonald's. McDonald's being the translational machinery. There's actually a study published by Hammerstrom et al. In 2020. It is called "The Benefits of Higher Resistance Training Volume Are Related to Ribosome Biogenesis." So there's a lot of debate in the field. How much volume do you need? Is more volume better for growth, for hypertrophy, etc.? Again, it's a debate. There's no consensus, whatever. But this specific study suggested that the relationship between your response to volume might be mediated by ribosomal biogenesis, which means the increase or the creation of new ribosomes. And ribosomes are the translational machinery. So essentially, it was saying that individuals with suboptimal adaptations to resistance training, if they increase volume, they may be seeing greater growth because of this increase of ribosome biogenesis. So it's kind of saying that the link between volume and growth, volume and size, volume and hypertrophy might be that more volume stimulates more creation of ribosomes, which increases your translational capacity, which allows you to increase MPS to a greater extent. The only kind of criticism that we have to the study is that it did compare 1 set versus 3 sets, and like, 3 sets is going to be better than 1 set. So I don't know if it's necessarily supporting a dose response relationship between volume and hypertrophy, because there's only 2 doses or 2 different volumes used in the study, and one was severely suboptimal, being 1 set. There's only so much we can say, but it's a good theory, and it helps us to kind of conceptualize what might be the link between volume and hypertrophy, if there is one. Let's talk about translation initiation. This is going to get confusing. It's going to get nitty gritty. It's going to get molecular, and I'm going to do my best to make it as digestible as possible. Translation initiation. It relies on a host of mRNA binding proteins that coordinate the recruitment of mRNA to ribosome. Again, mRNA is that gene that was transcribed into RNA that is ready to be processed by a ribosome, which is the translational machinery. That ribosome is what turns mRNA into a protein, okay? So translation initiation relies on a bunch of different key players, key proteins that are going to essentially bring that mRNA to the ribosomes, allow them to come together in the way that they're supposed to for translation to occur, and for those proteins to be created from that mRNA. The initiation of translation is the binding of mRNA to the 40S subunit of the ribosome. If you are a note taker when you listen to my podcasts, this is where I would say, get your notebook out. I am not going to cover every single little step of this process, because I do think that without visuals, without a proper lecture, you are going to get so lost. Like, there is so much going on. And like I said, I'm going to do my absolute best to make this as digestible as possible, while still ensuring that you guys are able to identify the most important key players that you may come across in research that you're reading. Okay. So there's 2 parallel processes that are occurring in order for translation to be initiated. There's a subunit of the ribosome. It's called the 40S subunit, and that needs to come together with mRNA in order to initiate translation. And in order for this 40S subunit to come together with mRNA, it is mediated by eukaryotic initiation factor 4F. So EIF 4F. This 4F initiation factor is a complex made up of 3 other subunits. We're not going to get into what those specific subunits do, but it's essentially EIF4A, EIF4G, EIF4E. So you need A, G, and E to come together, and that's going to create the complex that is initiation factor 4F. Okay, think of F as family. They've all come together, so it's a family. That's the one that we need as a fully cohesive, functional complex in order for the mRNA to come together with that 40S subunit of the ribosome. There is an inhibitory protein, and this is one that you will probably come across in research. It's called 4EBP1. 4EBP1 blocks the formation of this complex. So it actually inhibits a binding unit on the EIF4G subunit that prevents it from binding to EIF4E. You don't really need to know that, but if you want the specifics, that's what it is. So it binds to EIF4G, prevents it from binding to EIF4E, and then that prevents the whole complex from coming together and forming EIF4F. So in biology, we talk about phosphorylation a lot. If a protein gets phosphorylated, it usually gets either activated or inactivated. Phosphorylation is what we call a biological switch. It either turns things on or off. So 4EBP1 is this quote unquote bad guy. It's inhibiting one of the subunits of this EIF4F complex that we need intact. We need it all together. And 4EBP1 is preventing that complex from coming together. However, when 4EBP1 is phosphorylated, it is inhibited. So when you inhibit 4EBP1, that complex can fully come together and form because 4EBP1 is phosphorylated. It's inhibited. It cannot block those subunits from coming together. That's its normal instinct. It's kind of like the phosphorylation of 4EBP1 is like you're putting handcuffs on it. That 4EBP1 wants to come and snatch some of those subunits and prevent them from binding all together and creating the 4F complex. And phosphorylating 4EBP1 is like putting handcuffs on it. And it's saying, nope, like you're in jail, like jail time for you. You're done. And 4F is going to come together so that it can help initiate translation. Okay. So what phosphorylates 4EBP1? mTOR. So I know when you read research, you're going to come across mTOR levels were increased, mTOR phosphorylation levels were increased, this, that, and the other. mTOR phosphorylation means that mTOR is activated. mTOR being present means that mTOR can phosphorylate 4EBP1. So if you see increases in mTOR, that means 4EBP1 is being phosphorylated, it's being handcuffed, which means that this EIF4F initiation factor complex can form and allow that mRNA to be brought to the 40S subunit of the ribosome, and translation can initiate, which means mRNA can be converted to protein, meaning protein synthesis levels are going to be upregulated. So let me repeat that. If mTOR is present or mTOR is upregulated, or you see in a study increased levels of mTOR phosphorylation, you know mTOR is phosphorylating 4EBP1, meaning it's inhibiting that, meaning it's preventing that 4EBP1 from interrupting this complex that we need formed from forming. So if 4EBP1 is phosphorylated, it's inhibited, meaning the EIF4F complex comes together and aids in bringing the mRNA to the ribosome, increase in translation, meaning increase in muscle protein synthesis. Okay. There's another thing that needs to occur. So that is part of translation initiation. The other part is a complex called EIF2, eukaryotic initiation factor 2. This is a GTP-dependent process, and what it does is it binds tRNA, and the binding of EIF2 with this GTP and this MET tRNA forms this ternary complex. And this complex binds that 40S subunit along with other factors, and that forms the 43S pre-initiation complex. The only thing you really need to know there is that once EIF4F, so that family, that complex, binds the mRNA and brings it to the 40S subunit, you also need EIF2 to bind tRNA, bring it to that 40S complex. It requires GTP. And once those things come together, it forms 43S, which is another pre-initiation complex of translation. So EIF4F plus mRNA plus the 40S subunit is met or is joined by EIF2 with GTP and tRNA, and those things together form the 43S pre-initiation complex. And that is a ribosomal complex. So we are almost there at translation being underway and at protein being produced. Once you have that entire complex, remember GTP is part of it. GTP is going to hydrolyze, meaning it's going to go from GTP to GDP. It's going to release that EIF2, and then that is going to bind the 60S subunit of a ribosome, which is the large subunit of a ribosome. And that is where you have the full complex together. So you have mRNA being brought to the small subunit, 40S, of the ribosome. In order for that to occur, you need EIF4F as a full complex to help aid that binding. And in order for that complex to be formed, you need 4EBP1 to be phosphorylated, aka inhibited, aka handcuffed by mTOR. So once EIF4F and mRNA are brought together to the 40S, it's going to be joined by initiation factor 2, EIF2, GTP, and metRNA. Once all of that is together, GTP is going to hydrolyze into GDP. It's going to release that initiation factor 2, and that complex can then bind the 60S large ribosomal subunit, and then you're ready to start translation. I know this is a lot, so if you need to rewind, take notes, try to follow along, find a diagram, I really recommend that. I'm going to link some diagrams for you to follow along in the description. But yes, so the kind of last key players we're going to talk about is we're going to go back to mTOR, because mTOR does a little bit more than just phosphorylate 4-ABP1. mTOR is a key mediator of hypertrophy. It's probably the most abundant protein that you're going to read about when you're reading hypertrophic research that does muscle biopsies, that looks at biomarkers, etc. Many studies will note an increase in mTOR phosphorylation in response to resistance training, in response to muscle loading. And the primary action of mTOR, we talked about how it inhibits 4-ABP1. It has another parallel function, which is via a protein called S6K. And you will read about S6K or S6K1 levels or S6K phosphorylation levels in research as well. And a lot of times, if you see a study report increases in S6K phosphorylation, they're going to say that is indicative of increased mTOR levels, and therefore muscle hypertrophy is occurring. So S6K can be a proxy for mTOR upregulation, mTOR activation, etc. And we're going to go into how or why. But again, remember that the theory is that when you resistance train, you upregulate the transcription of mTOR, and mTOR in turn allows you to increase the translational rate of proteins needed for building muscle. Okay. The primary action of mTOR is to promote hypertrophy via S6K. So mTOR phosphorylates, we talked about how that's an on and off switch in biology. mTOR phosphorylates S6K, so in this case it's an on switch, and we know it phosphorylates 4-ABP1. We already talked about that. When it comes to S6K phosphorylation, like I said, this can be a proxy for mTOR activity. So if you see increases in S6K phosphorylation, you know that's an increase in mTOR activation and mTOR activity, because mTOR is what's phosphorylating that S6K. S6K is responsible for RPS6, which is ribosomal protein S6, which is a part of that 40S subunit that we talked about previously. And essentially, all you need to know is that a fully activated S6K protein phosphorylates a number of initiation factors that promote the formation of this initiation complex. And it also is involved in the elongation process. So not just initiating translation, but keeping it going. And so basically what you need to know is that mTOR phosphorylates, like we said, 4-ABP1, which is going to allow that translation initiation complex to come together. But it's also going to phosphorylate S6K. And S6K is going to phosphorylate other initiation factors that are going to, in turn, also help increase the rate of translation by activating those initiation factors needed to create that initiation complex. So it does the same thing via two different pathways, two parallel pathways. By phosphorylating 4-BP1 and by phosphorylating S6K, mTOR is via both pathways parallelly, is that a word? Parallelly? It is increasing the rate of translation initiation. That's the biggest thing you need to know about. There was a 2007 paper done on blood flow restriction, BFR, and it showed increases in S6K1 phosphorylation. And it also looked at MPS rates, so fractional synthetic rates, which I believe is mixed. It means muscle protein synthesis, both myofibrillar and mitochondrial. And it did find that those increased by 46%, but it also found that S6K1 phosphorylation increased. And the major finding was that the downstream protein of mTOR was stimulated and muscle protein synthesis increased following this low load blood flow restriction training program. This was kind of the foundational paper that suggested that BFR did have molecular support for muscle growth outcomes. So the muscle growth outcomes that had been previously seen by BFR had never been supported on a molecular level, and this kind of gave the molecular corroboration of like, yes, we're seeing this muscle growth happening in response to blood flow restriction training because we're seeing an increase in muscle protein synthesis, but also an increase in that S6K1 phosphorylation, which is a proxy for mTOR activation. This other kind of study done on transgenic mouse models, it kind of knocked out mTOR, and the mice did not see any hypertrophy. So that was just kind of confirmation that mTOR kinase activity is both necessary and sufficient to induce hypertrophy. Now, that one was done in mice. So huge moral of the story, if we're going through everything, it is that a bunch of different key players need to come together in order to initiate the translation process, which is the conversion of mRNA into functional proteins. For that process to be initiated, you need to first have the mRNA bind to the 40S subunit, which requires the assistance of the EIF4F initiation complex. That complex cannot be formed until 4-ABP1 is phosphorylated, aka handcuffed, by mTOR. Once that entire complex comes together, it is joined by EIF2, GTP, and MET tRNA. That is the 43S pre-initiation complex. Then GTP is going to hydrolyze to GDP. It's going to release that EIF2, and that can now go and bind to the large 60S ribosomal subunit, which is now the completed picture of translation initiation, and you can go on and actually translate that RNA into proteins. And the rate at which all of those key players can come together, so the rate at which you phosphorylate 4-ABP1, or you phosphorylate S6K1, or you bring the mRNA to the 40S, or you hydrolyze GTP to GDP, the rate at which any one of these little steps in this entire complicated process are happening, if you can increase the rates of one or two or all of those things, you increase the rate at which translation is initiated and therefore increase the rate at which protein is produced. So you upregulate your muscle protein synthesis rates if you can increase the rate of any or all of the different moving parts of this initiation situation that I've just described. So increasing the rate of all of this happening is what we call an increase in translational efficiency. So it's essentially increasing the translation of specific mRNA without increasing the amount of mRNA or without increasing the amount of translational machinery, so the ribosomes. It's just saying you have the same amount of starting product and you have the same amount of machinery, you're just getting it to occur faster. So resistance training in rats showed that the rate of muscle protein synthesis per unit of RNA increased in response to resistance training. So this is animal research, animal model support, that resistance training does increase the rate of translation, independent of an increase in RNA or an increase in ribosomes. This indicates, like I said, an increase in translational efficiency. So the rate of addition of peptides, which are the building blocks of protein, has the potential to increase. And this primary mechanism of that increase is thought to involve initiation, which is everything that we've talked about. There's other things that I'm not going to kind of get into, but that's essentially translational efficiency. That's one of the ways in which we upregulate muscle protein synthesis. The other one is translational capacity, which is essentially you can increase the rate at which that translational machinery is working. So those ribosomes, you can increase the rate at which those are working, but there's a limit. And if you only have so many ribosomes, there's eventually going to be a cap to your rates. But if you increase the number of ribosomes, you increase your overall translational capacity. Or on the other hand, there's only so much DNA, so there's only so much that you can transcribe and translate at a time. So the primary limitation might be the bulk amount of DNA present in a given myofiber. So when we talk about translational capacity, again, we can increase it either by increasing the amount of mRNA, by increasing the amount of DNA, or we can increase it via the amount of ribosomes. I don't know if you've ever looked at a diagram of hypertrophy at a muscle cell level, but a lot of times we talk about the increase of myonuclei in a muscle fiber. So satellite cells donate their nuclei to a muscle fiber and they fuse, and then you get what's called a multinucleated cell. So muscle cells are multinucleated. And more myonuclei, so the more nuclei that your muscle cell has, the more DNA it has, which means the more capacity for translation it has. So when we're talking about the production of proteins, it might not be particularly critical to have all these nuclei because the translation of mRNA is subject to amplification. So when that RNA polymerase transcribes DNA into mRNA, it can do that a lot. And then you can also have polyribosomes, so multiple ribosomes on one mRNA kind of going all at the same time, kind of like a production line in a factory. But the number of copies of rRNA, which is the RNA that encodes for the ribosomes themselves, or tRNA, which is part of that initiation complex. It's what's actually involved in bringing the anticodon to the codon and then actually bringing the amino acids to the codons. That's beyond the scope of this lecture, but point is, they're two critical parts in translation. The number of copies of those can only really be increased via the acquisition of additional copies of template DNA. So the benefit to having more nuclei in your muscle cell is actually so you can create more rRNA and more tRNA, and therefore increase your ribosomes and your tRNA and increase your translational capacity. So it's not so much that we want more DNA so we can get more of the protein product genes. It's so that we can get more of the machinery genes, the genes that actually encode for the machinery that we need for translation. And the most likely source of these nuclei that help increase this amount of template DNA is the satellite cells. And we have evidence that this general increase in efficiency occurs at the onset of muscle hypertrophy. And sustained increases in protein production appear to require substantial increase in translational machinery. So again, if we want to produce protein at greater rates over a long period of time, that's not going to occur just from getting things to happen faster. It's going to occur from having increased capacity to do more at a time. So more ribosomes, more tRNA. And there's a study, again, done in rats, where they were subjected to synergistic ablation, which is where you essentially cut out a synergistic muscle. So think if you're walking and you just remove part of your quad, the other parts of your quad are going to need to now sustain your entire body weight and movement and everything. And they're probably going to grow as a compensatory response for now needing to sustain everything. So they did that in these rats, and there was a 30% increase in the RNA to DNA ratio after 15 days. So in the first 15 days, we did not see that increase in DNA. We did not see that increase in nuclei, but we did see an increase in RNA, meaning an increase in the rate, in the efficiency. However, after 90 days, the RNA increased, but so did the DNA. And that ratio went back to original. So you got that increase in efficiency at first, and then over time, you got more nuclei, therefore more DNA, and therefore more translational capacity. So we do have animal model research suggesting that the time course of muscle protein synthesis rate upregulation is first at the efficiency level. So you increase the rate of translation initiation, and then over time, chronically, you actually increase your overall translational capacity. So that's pretty much what I have for today. And I know that was a lot. So if you guys need a refresher, questions, just didn't understand, this was probably the most technical episode I've done yet. And I really underestimated how hard it would be to explain everything without like visuals, and how hard it would be to explain everything like with recognizing that I sat through this lecture. It's not the first lecture that I've discussed these molecules. I have like a molecular background. So I don't know if any of this made sense, to be honest, but I really, really, really hope it did at least a little bit. And the overall takeaway is that when we're talking about the molecular response to resistance training, we know that it starts with lifting a weight and it ends with an increase in muscle protein synthesis. And what happens between that, how we increase those rates of muscle protein synthesis, is essentially having these key molecular players that need to increase their activity so that translation, or the conversion of RNA into protein, can occur at a greater rate. And if we're talking about acute or short-term adaptations, that rate is going to increase. So translation initiation, that rate, it's just going to increase. But if we're talking about chronic adaptations and chronic responses and long-term muscle protein synthesis rate upregulations, then we're not just talking about increasing the rate, we're talking about increasing the overall amount of translational machinery of ribosomes that we have. If I had to oversimplify this, it's going to go from lifting the weight, so doing the rep, so one bicep curl, not one, but you get the point. Bicep curl upregulates mTOR. mTOR phosphorylates 4-ABP1. That inhibits it. It allows this EIF4F complex to come together. That allows mRNA to bind the 40S subunit. You have EIF2. That's going to bind GTP, MET tRNA, and come together with the mRNA, EIF4F, and 40S subunit to create the 43S pre-initiation complex. GTP is going to hydrolyze into GDP, release that EIF2, and that is going to bind the 60S subunit and allow translation to be initiated. At the same time, mTOR is not just phosphorylating 4-ABP1, but it's also phosphorylating S6K. S6K, now phosphorylated, is going to phosphorylate RPS6 along with other initiation factors that is going to allow that entire initiation complex to just happen more efficiently. So again, you lift, you increase the transcription and the translation of mTOR. So you increase your mTOR levels and your mTOR activation and your mTOR activity. And that, via two parallel pathways, is going to essentially kind of create a domino effect of different steps along the signaling pathway that's going to allow this entire translation initiation complex to occur faster, more efficiently, and over time at a greater capacity. So with that, I hope that recap helped. I hope this was interesting. Like I said, I'm a molecular girly through and through. So for all my molecular queens and kings, I hope you enjoyed this. But otherwise, if you have any questions, please email thevaliditypodcast@gmail.com or DM my podcast Instagram or my personal Instagram, VallFitt_IFBB Pro. And if you're interested in participating in the world's first reverse diet study, please email reversedietstudy@gmail.com. I am taking one-on-one clients now that prep is done. I have a little bit of increased capacity, so to say. Since we're talking about increased capacity, I'll be a ribosome for the day. No, but my new application, because I had issues with the old one, is also linked in the description. So if you do want to work together, I do video responses and we do get into science anytime you have questions. If you like this, then I think you'll enjoy that part of the check-in process, amongst other things. But yeah, so as always, I hope you guys enjoyed. Let me know if you have questions, and I will see you in the next episode.